Hello, everyone. Welcome to the Target 2035 webinar series. And today I'm happy to introduce Ingrid Wirtz, who is going to be chairing our session on targeting deubiquitinases for therapeutic benefit. And so Ingrid is the Executive Director of Protein Homeostasis Center of Excellence at Bristol Myers Squibb. She and her team are responsible for developing protein degrader medicines for patients in need by harnessing degradation systems naturally present within the human body. Their aim is to specifically target and eliminate disease-relevant proteins that have been largely undruggable in the past. Prior to joining Bristol Myers Squibb, Ingrid was a principal scientist in the Department of Discovery Oncology Early Discovery Biochemistry at Genentech, where she led the company's degrader platform aimed at co-opting ubiquitin system enzymes for therapeutic benefit. So it's my pleasure to welcome Ingrid, who's going to chair and lead this session. Ingrid.
Thank you very much, Cheryl. So I will share my screen now. Okay, can you see the screen?
Yes.
Okay, excellent. So thank you very much, Cheryl. Thanks to the organizers for allowing us and inviting us to present this webinar on targeting deubiquitinases for therapeutic benefit. And I'm really pleased to be here with Sarah Berlage and Anthony Varka. So Sarah is an associate professor of biological chemistry and molecular pharmacology at the Dana-Farber Institute. And her chemical biology lab really established an integrated deubiquitinase-focused platform with the goal of developing selective probes and annotating the function of deubiquitinases and the translational potential for deubiquitinases in disease. And Anthony just graduated from the Harvard Chemical Biology PhD program in Sarah's lab, and excitingly just took a position at Therapeutics. And his research focused on designing and implementing high-throughput screens in order to identify small molecule deubiquitinase inhibitors and probe compounds to study DUB functions in disease. So really pleased to be here with Sarah and Anthony for this webinar. So we'll just begin with a brief introduction by myself, and then Sarah and Anthony will talk about the science of targeting deubiquitinases for therapeutic benefit, and then we'll move to audience Q&A. So just by introduction, I'll review the protein homeostasis in cells as regulated by the ubiquitin system. So really this all starts with an assembled ubiquitin ligase. This can be multi-subunit complex or a single protein ubiquitin ligase. And that ligase is then competent to recruit a target protein for ubiquitination. And so ubiquitin is a small protein that's covalently ligated to the target protein. And since ubiquitin itself is a protein, multiple rounds of ubiquitination can then result in polyubiquitin chains. And then often these polyubiquitin chains can serve as a tag to bring the ubiquitinated protein to the proteasome, because the proteasome has ubiquitin binding subunits on it. And the proteasome is a multi-subunit complex. It has proteases and unfoldases. So the target is unfolded, threaded into the proteasome core, and then targeted for proteolysis and subsequent degradation. And as with many key cellular processes, ubiquitination can be reversed by deubiquitination, and that is achieved by deubiquitinase enzymes, or DUBs. So I mentioned that ubiquitin is a protein itself, and it has multiple lysine residues. And when these lysine residues are ubiquitinated, this can form polyubiquitin chains. And when these chains are often linked by lysine 48 or lysine or branch chains thereof, typically this serves as a tag to target the protein for proteasomal degradation. And so when deubiquitinases remove these chains and return those chains into, depolymerize them and change, basically move them into the free ubiquitin pool, this can really result in rescuing targets from degradation. And when polyubiquitin chains are assembled into lysine 63 link chains or linear ubiquitin chains, these types of chains often assemble signaling complexes. And deubiquitinases can disassemble those and to result in the disassembly of the complexes themselves. As for free polyubiquitin chains, those can be disassembled by deubiquitinases as well. And finally, ubiquitin itself is transcribed and translated into precursor proteins. Deubiquitinases can cleave those chains and release free ubiquitin from these precursor proteins, again, to contribute to the free ubiquitin pool. So you can see that deubiquitinases are really critical for regulating essential cellular processes. Deubiquitinases also come in two enzymatic forms, the cysteine proteases and the metalloproteases. As for the cysteine proteases, what we've listed here is the family name, and so you can see that there's 6 cysteine protease deubiquitinase families, and then the number of deubiquitinases within each family. And then we're also showing an exemplar structure of each family class along with the PDB code. As for metalloproteases, there's the JAM metalloproteases with CSN5 as the exemplar deubiquitinase there. And so importantly, deubiquitinases are also conserved in vertebrates, in yeast, in plants, and beyond that, microorganisms, for example, bacteria and viruses, encode deubiquitinases, and they also co-opt the host ubiquitin system to enhance their pathogenesis. So overall, we've reviewed that deubiquitinases regulate essential cellular processes. We've shown that humans encode approximately 100 deubiquitinases that fall within 7 different families. Again, these are either cysteine protease or metalloprotease enzymes. And so collectively, we believe that deubiquitinases are very attractive therapeutic targets. And indeed, potent and structurally defined deubiquitinase inhibitors have been identified, and these inhibitors are progressing through clinical development. And so we'd like to emphasize that really with any emerging therapeutic ambition, collaborations will really be essential to expeditiously benefit patients in need that have diseases that can be treated with regulating deubiquitinases. And so with that, I'd like to turn it over to Sarah and Anthony, who will be talking more about targeting deubiquitinases for therapeutic benefit. Thank you.
Excellent. Well, thank you, Ingrid, and all the organizers for the invitation and the opportunity for Anthony and I to share some of our work. If we could just ask you to stop sharing, then we will bring up our slides. Great. So I just have a couple slide intro here, big picture, about the way my lab is thinking about deubiquinase enzymes, and then I'm going to hand it over to Anthony. But Ingrid very nicely set the stage for where DUBs are at. They're really, an emergent class of drug targets right now. As was noted, the first agents are now entering the clinic. We know that they are chemically tractable, and in fact, there are multiple pockets, both for inhibitor, activator, and recruiter development, although our talk today will focus on the inhibitors. Just to summarize it succinctly, I think about, DUBs as kinases 20 years ago. So next slide, please, Anthony. And so, one of the reasons I was really excited to really get into the DUB field was because of the tremendous promise and I think impact that I felt that my lab could have taking a chemistry-centric approach to the deubiquinase enzymes. And so really in our programs, we implement chemical matter identification quite early. So we can utilize this really to synergize with target biology to advance both aspects of a project, with the goal at the end that we're now going to have a compound for a given DUB. It's really going to enable us in understanding the substrates, the basic function, and then we can look at the translational potential, when appropriate in our lab, and often also, through collaborations with our colleagues at Dana-Farber and elsewhere. And so just next slide, when the lab started, we implemented this approach first, starting for one dub, USP7, which is one of the most well-studied dubs across both academic as well as, industrial research groups. In our lab, we've reported several generations of probe, and we've done a number of studies to look at unbiased fashion at function, as well as focus studies with collaborators on disease relevance. And so the work from our lab has shown that inhibition of USP7 can promote cancer cell death directly through upregulating P53. And excitingly, in some instances, also can sensitize cancer cells to immune surveillance, and this is through a role in regulation of a non-canonical polycomb repressive complex 1.1. And so this was, a really great first project for us to really start to build capabilities, assays, learn more about the dub field. But we really want to think about dubs as a target class. There are 100 of them, and we don't want to work on them one at a time. We want to work on many in parallel. So that really meant thinking carefully about some efforts to start to fill the chemical matter pipeline with a wide array of different starting points, as well as efforts to really increase the number of target hypotheses that we had high confidence in. So today, Anthony's going to talk about some of the work really focused on expanding the chemical starting points and probes for a wider array of dubs. So I will just show one more slide then that is a plug for a recent paper that just came out that was focused more on the target biology. And this really was a multi-omics approach where we generated some, new data, and we integrated it with the vast amount of data that is, publicly available to really start to drive either new hypotheses or help maybe clarify which hypotheses in the literature, we might want to follow up on first. And for those of you particularly interested in dubs, we have a dub portal website that is easily searchable to see the, kind of collated data that we have for a given dub of interest. And so with that, I will hand it over to Anthony to really tell you about our efforts on the small molecule discovery piece.
Yeah, so thank you for the opportunity to come today and present some of the research that I've done in Sarah's lab on the discovery and development of novel dub inhibitors through parallel high-throughput screening. And so just to quickly recap, as was pointed out earlier, deubiquitinases or dubs are cysteine proteases primarily that cleave ubiquitin from protein substrates and break down ubiquitin polyubiquitin chains. And so here again, I'm just showing a breakdown of the different families based on active site sequence homology, as well as representative structures for each of the different families. And so when I joined the Burlage lab, we really looked at a survey of kind of the literature at the time. This is a review from 2018 that showed that there were some dub inhibitors that had been reported as single dub inhibitors. And so for example, for USP7, this HBX compound that I'm highlighting here was discovered and reported as a USP7 inhibitor. And so some of the downfall of this early kind of dub inhibitor development is that while compounds were found for different dubs, they weren't really characterized for selectivity amongst all of the different dubs that exist. And so if we look at a broader profiling experiment, so this is a diubiquitin cleavage assay that has a readout by mass spec. And so basically in this paper, what they did was took different dub inhibitors that had been reported and essentially asked, what is the inhibitory profile against a large panel of deubiquitinases? And if you actually look, this HBX compound that was reported as a USP7 inhibitor was actually profiled. And here what we can see is that, darker red indicates a higher percent inhibition. And so this compound, while, does have some activity against USP7, it also hits a number of other dubs, which really reduces its utility as a probe compound to really understand the dub function. And so primarily, a lot of the time, these compounds are found through high throughput screening. And so you start with your small molecule library, often, over 100,000 compounds or more. You have your protein of interest, some assay to assess, and then ideally, when you do all of this, at the end, you'll get out screening hits that can then be optimized for potency and other properties that are really desired. And so when I joined the lab, we had actually just begun a collaboration with the Novartis Institutes for Biomedical Research across the river in Cambridge. And essentially, they have a 50,000 compound library comprised of synthetic small molecules and some natural products that they make available for academic collaborations. And so they have the screening platform, the compounds, you bring your targets, and then you can use kind of the setup there to test their compounds against whatever target you're interested in. And so when we were thinking about, how can we best leverage these compounds, this setup to really get not only potent DUB inhibitors, but also compounds that are selective so that we don't have to engineer selectivity through medicinal chemistry efforts. And so we really wanted to focus on kind of incorporating selectivity within the screening process. And so what we decided to do was to screen multiple DUBs against the same 50,000 compound library. And so the idea here is that each DUB is seeing each compound in the library, and hopefully out of that, we would be able to find compounds that were hitting single DUBs over all of them that were included. And so for our selection criteria, we really focused on, can we get a scalable quantity to actually do the high throughput screening? That was obviously one of the first things. But then we wanted to think about, what DUBs are associated with interesting biology in the literature that if we got hits, we could follow up on. And we also wanted to think about, can we get diversity within and amongst the different DUB families that exist? And so that leads me into the first part of my talk, which will be the screen that we did in collaboration with Novartis. I'll talk about kind of the design and implementation, as well as some of the results from that. And then I really want to focus on the medicinal chemistry efforts for one of the series that came out of that screen against USP28. And so just to give you a quick overview of how we kind of set up our high throughput screen. So I'm showing on the phylogenetic tree on the right, highlighted in red arrows, the 8 different DUBs that we ended up screening. And so again, these were selected based on the number of criteria that I mentioned previously. But one of the important things that I'll point out here, again, kind of looking at this tree, is that we really wanted selectivity within and amongst the different families. So we got 6 representative members within the largest family, the USP family, as well as members from the UCH and OTU families as well. So it's not perfect, but we tried to get as much kind of representation as we could. And so once we had our targets, we proceeded to assay optimization. So this included some buffer screening, miniaturization, as well as enzyme and substrate titrations to be able to really optimize the conditions for each compound. And so obviously for doing high throughput screening, you need some sort of assay. And so the assay we chose is a ubiquitin rhodamine assay. So it's a very simple purified enzyme assay in which we incubate our purified enzyme or DUBs with a ubiquitin rhodamine substrate. So this is kind of mimicking the natural cleavage point of diubiquitin or multi-ubiquitin polymers. And then we have our compounds or DMSO present. And so the idea is that in the absence of compound, or if your compounds cannot bind, then your DUB will bind ubiquitin rhodamine substrate, cleaving off the rhodamine, which can then be read out by a fluorescence plate reader. And so if your compound does bind and inhibit, it will prevent the cleavage of this, and this leads to reduced fluorescence output. And so by using this kind of surrogate assay, we can really get a sense of how the compounds are interfering with the ability of DUB to process ubiquitin. And so after we had kind of optimized the conditions for each DUB, we proceeded to the primary screen. And so the primary screen was just a single concentration of each inhibitor against each of the 8 DUBs run separately. So each DUB was screened against all approximately 50,000 compounds. And then we took a look at the hits from that primary screen. Those were reordered in dose response. And then we retested those compounds in dose response against the target and at least one of the other DUBs included in the screen. And so this was again to get a sense of selectivity as we were doing our dose response confirmation. And then from there, we really prioritized some of the hits that looked really potent and selective for additional validation experiments. And so I want to talk now about the results from the primary screen. So what I'm summarizing here is just a breakdown of all of the compounds in the library against each of the different DUBs. And so just to orient you on how to read this upset plot, if we look at our first column here that has about 1,400 compounds in it, and you follow that bar down to the bottom, you'll see there's a single dot filled in that corresponds to USP17. So what that means were these compounds were hits in the primary screen for USP17 and only USP17. And so I'll say here that for most of the DUBs, the compounds were screened at 25 micromolar. For USP17, we actually screened them at 50. And we used a very modest hit calling here. So hits were called as having at least 30% inhibition. And so that's very modest, but we really wanted to kind of consider as many compounds as we could because we knew we were going to follow up in dose response. And so I've highlighted in red all of the individual bins of compounds that were selective for individual DUBs. And so I'm only showing the top 40 clusters here, but you can appreciate that all 8 are included in kind of the top 40 bins, but they're not exactly distributed evenly. Some have, hundreds to thousands, whereas others are down to the, tens to 12 range. All of the other bars that are filled in black represent compounds that scored as hits against more than one DUB. And so those DUBs are kind of highlighted in for each filled in dot. And so while the design of this experiment and this high throughput screening was really to focus on selective inhibitors, there are interesting compounds here that might hit the USP family members over the others, or pan-dub inhibitors that could be interesting as tool compounds as well. So we've also been thinking about that. But as I mentioned, kind of the main goal of this was to identify selective compounds. And so we were really encouraged at this point that just in the primary screen, there was evidence that we were seeing selective inhibitors for the different dubs that were included. And so as I mentioned, the next thing that we did was we reordered all of those hits. So regardless of selectivity, as long as it hit a single dub, we reordered it in dose response and completed the same ubiquitin assay against each of the different dubs. And so again, here, we were screening against the target dub and at least one other dub that it was not a hit for. And so that was, again, to give us a sense of not only the dose response confirmation, but also the selectivity for the dub that it scored as a hit for. And so in this kind of circular pie chart, I'm just summarizing the hits from the dose response, as well as those hits filtered through a couple of different criteria. And so I just want to take a second to walk through each of these circles. So if we start with this first outer ring, these were compounds in the dose response that scored as a hit for just the target. And so for calling a hit, we're just looking at below 40 micromolar, or in the case of UCHL1, the highest tested dose was 25 micromolar. So below essentially the highest tested dose for each of the dubs. And then at least 30% inhibition of the target. So again, these are very weak criteria for calling hits, but they kind of mimic the primary screen results. The next thing that I did was apply what I'm calling selectivity filter. So now I'm excluding all of the compounds that essentially had an IC50 against the non-target dubs that they were counter-screened against. And so hopefully what you can appreciate here by just looking at the numbers is that this kind of very modest selectivity filter actually excludes quite a number of compounds from consideration, which kind of again points to the fact that thinking about selectivity, especially within a large family such as dubs, is quite important when going through this process. The next filter that I applied is kind of the first potency filter. So now again, we're looking at kind of within these selective compounds, decreasing the IC50 against the target to 10 micromolar or below. And then the next filter is kind of getting to our most potent compounds, which is keeping that below 10 micromolar against the target, and now increasing the max inhibition of the target dub to at least 80%. And so what you can appreciate here within this kind of middle circle in black is that for most dubs, we actually had no qualifying hits. For a couple, we only had 1 or 2, and then USP28 was the clear standout with 74. If you actually look at the structure of these 74 hits, we actually noticed that there were quite a number of compounds that shared the same core scaffold, but with just slight modifications. And so this gave us kind of some evidence and some initial SAR that these were real, actual hits against USP28. But in order to figure out, where do we prioritize our follow-up efforts, we looked at the distribution of IC50s for these most potent hits for each of the dubs. And so what we saw was that, for USP28, we had quite a few, and there were actually some that were, very potent in the kind of sub-micromolar range. And then the lone USP30 hit was also quite potent. And so we really focused on these 2 dubs to prioritize for follow-up. And so the first thing that we did was just resynthesize each of the compounds in-house and then test them in dose response in our ubiquitin assay. And so I'm showing the results here for those resyntheses and retests. And we saw that for USP28, the compounds that we resynthesized showed really nice activity that was in line with what we were seeing in the dose response at Novartis. And again, for USP30, we resynthesized the USP30 hit in-house, retested it, and saw really good confirmation with the activity that we were expecting. And so the next thing that we wanted to do was get a sense of what's the selectivity for the target dub over a broader panel of dubs. And so we sent our compounds to Ubiquigent, which is a commercial service that tests compounds against a larger panel of dubs using a similar ubiquitin rhodamine assay. And so for this, the USP28 inhibitors that we sent showed really strong inhibition of USP28. So in here, the heat map, darker red indicates a lower % of the dub activity remaining following compound treatment. And this is compound treated at just a single concentration. And so for our USP28 inhibitors, we saw really potent and strong inhibition of USP28. And we did see one other off target, which was USP25. This actually isn't that surprising. In the literature, USP25 and 28 are very highly homologous to one another, even within the USP family. They share a unique insert within their catalytic domain that kind of set them apart from a lot of the other USPs. And there actually is some precedent in the literature for kind of dual 25-28 inhibitors. But beyond that, we saw really good selectivity for just these two dubs over all the other dubs that are included in this panel. And then for our USP30 inhibitor, we saw really nice potent inhibition of USP30 and really no additional off targets in all of the other dubs included in the panel. And so then we proceeded to another validation experiment. And so this time we used our activity-based protein profiling assay. And so in this experiment, we take cells and lyse them and then treat with either inhibitor or DMSO. And so the compound in cell lysate will bind to any dub that it's able to. And we followed treatment then with a activity-based probe. And so this contains ubiquitin that has an electrophile as well as a biotin tag. And so in cell lysate, the electrophile will covalently label all of the catalytic cysteine residues of any dubs that the ubiquitin probe can bind to, but it will be unable to label any dubs that are inhibited by the probe. Sorry. And then we follow this with a streptavidin pull down to enrich for all of the dubs within our sample or anything bound by our ubiquitin probe. We then trypsin digest, TMT label, and do LCMS-MS to get a sense of which dubs are present and absent in the compound and DMSO treated samples. And so essentially, the heat map I'm showing at the bottom is a result of this experiment. And so the darker red indicates a higher percent blockage of probe labeling, indicating that our compound is binding to those dubs and preventing the probe from being able to bind and label. And so for our USP28 inhibitors, we saw really good inhibition of USP28 for two of the more potent ones, AV9606-180 and BIN010707. And then for the rest that were slightly weaker, we did see activity against USP25. We included AZ1, which was a literature reported kind of the best in class at the time, 25-28 inhibitor. And for that compound, we do see activity against 25, not as much against 28. I will say that in this experiment, since the probe is covalently labeling, whereas all of these compounds are non-covalent, we often see kind of an underrepresentation of our inhibitors. So as the compounds bind, they can dissociate, but once the probe goes on, it stays on. So it often makes non-covalent compounds appear a little bit weaker. But we were encouraged to see that for our more potent ones, we were getting activity against 28 as well as 25. And we saw no additional off targets across any of the other dubs that were detected in this experiment, indicating our compounds are only binding to those two dubs. For the USP30 inhibitor, AV11324-5, we again see really nice activity against USP30 and no other additional off targets. We also included XL188, which was our the USP7 inhibitor developed in the lab just as a positive control for the experiment, just to make sure that we were seeing what we expect to with that compound. And so we went a little bit further to profile some of these compounds. And so I'm showing the results here of our target engagement experiment. So it has the same setup as the activity-based protein profiling I described previously, except that instead of reading out by LCMS, we read out by Western blot. And so the idea here is that if your inhibitor binds and prevents the ubiquitin probe from binding, you'll only see this lower molecular weight adduct, which represents the native dub. And so this will be a band that appears lower in the gel. And if your compound does not bind or cannot compete with the probe, you'll get labeling with this ubiquitin probe that results in a higher molecular weight species and leads to a shift up in the bands that we're seeing in the Western blot. And so the top plot that I'm showing you here represents some of our more potent USP28 inhibitors in dose response. So from 100 micromolar down to 1 micromolar. And what we see here is that for the higher concentrations, even down to around 10 micromolar, we're really only seeing the lower molecular weight band. In some cases, you're starting to see a little bit of the higher molecular weight one as well appear as a shadow on top. And so, as we get down into the 10 to 1 micromolar range, we're really kind of still getting a little bit of competition with the probe, and you're starting to see both bands. But this really indicates that even at concentrations down to the 1 to 10 micromolar range in cell lysate, we are able to effectively compete with this covalent probe. The bottom blot that I'm showing you represents some of the lower or higher IC50 compounds, so weaker inhibitors in the same experiment. And as you can see, commiserately, we see kind of a shift to a higher concentration needed to be able to start to effectively compete with the probes. And so we were really excited by these USP28 inhibitors. They looked really good in target engagement. We did have this really potent USP30 inhibitor that we had identified from all of our screening efforts. We had demonstrated by Ubiquigent profiling in ABPP that it was selective for USP30 amongst all the other dubs. And so we were really excited to follow up on this hit as well. However, during the course of all of this work, another group actually published this scaffold as a selective USP30 inhibitor. And so while we were a little disappointed that we weren't able to publish this result, it really served as nice external validation that this approach was able to identify selective small molecule inhibitors. And so again, as I mentioned, kind of the main goal that we set out when we were doing this process was to be able to design a screening cascade that was able to identify selective small molecule inhibitors. And so having kind of an external validation that this approach worked actually was really nice to be able to kind of include in our results. And so just to quickly summarize this part of the talk, we successfully designed and deployed a high throughput screen with an emphasis on selectivity for deubiquitinases inhibitors. We identified numerous hit compounds for many of the dubs included within the screen. So I talked about USP28 and USP30, but there were a number of other dubs that had hits, some of which maybe were a little bit weaker, but still compounds that we were interested in following up on that we've been working on a little bit. And we've then taken, some of the more potent ones for follow-up selectivity profiling and validation experiments, which really kind of served to validate USP28, USP30 as dubs that had potent and selective inhibitors. And all of this work was published in Cell Chemical Biology, and our high-throughput screening protocol was actually also published in STAR protocols. And so now I want to move on to talking about some of the medicinal chemistry efforts around one of the series that came out of this Novartis screening effort. And before I do that, I really wanted to give just a quick overview of why we might be interested in developing probe compounds or inhibitors of USP28. And so USP28 is proposed to play a role in a number of different processes. It's been reported to antagonize a specific E3 ligase complex that uses this FBW7 adapter protein that recruits and ubiquitinates a bunch of transcription factors such as c-Myc, LSD1, and HIF1 alpha for proteasomal degradation. It's also been reported to play a role in the DNA damage response pathway. Looking at some of the USP28 targets, 53BP1 was one that stood out. It's been shown to actually interact directly with USP28 and reported to play a role in that interaction in the DNA damage response pathway. And so there's interesting implications for following up for DNA damage response. And it's also been shown to play a role in different cancers, such as in this paper, squamous cell lung carcinoma. And so there are a bunch of potential ways that we could follow up on the biology for having USP28 probe compounds. And so we were really excited based on that to really dig in and kind of identify some of the areas that were really driving compound activity. And so the series that we decided to focus on primarily was this BIN 010707 series, which represented one of the more potent hits from the screening efforts that validated well in our orthogonal validation assays. And so our initial focus was really on the two areas that I've highlighted here, and I just want to walk you through some of the key SAR. And so our screening hit BIN 010707, we retested that using a slightly higher throughput ubiquitin rhodamine assay, and we saw an IC50 for this compound of 122 nanomolar. And so the first thing that we looked at was this linker region, which I'm highlighting in blue. And so in our parent compound, we had an ethylene linker here, and we decided to try and reduce that to a single carbon, as well as extend it to 3 carbons. In both cases, we saw that we started to lose activity, indicating that we were really in the sweet spot with this kind of 2-carbon linker. We noticed that we had this NH group as part of our linker, and so we decided to try methylating it as well as replacing it with an oxygen. And in each case, both of these compounds, BIN 149 and 104, actually lost all activity, indicating that potentially this hydrogen on this nitrogen is involved in hydrogen bonding with the protein and could be really crucial for compound activity. We also explored some of the different tail pieces, so trying to replace this 3,4-dimethoxyphenyl group with different groups. Most of the changes we made in this region actually resulted in compounds with comparable or weaker activity, but we did start to see a slight improvement when we went to this kind of ore substituted with larger nitrogen-containing heterocycles. And so this BIN 170 compound represents one of the kind of advancements in all of that derivatization. We next turned our attention to the other end of the molecule. And so for this, we were really highlighting and thinking about this propyl chain as well as this cyclopentyl group. And so one of the first things that we did was just remove the propyl chain, and we saw that this compound lost quite a bit of activity. So now we're in kind of the 11 micromolar range. We then removed the cyclopentyl group and saw that we actually gained a little bit of activity back, indicating that that cyclopentyl might actually not be good for the observed compound activity. And so then we reintroduced the propyl chain and saw that we actually got a slight boost. So that is in keeping with kind of getting rid of that cyclopentyl. And that led us to a compound that was slightly more potent than the parent. And so then we really wanted to think about, since this propyl chain seems to be important, what are some of the size limitations? And so we made a derivative with the cyclopentyl on, but increasing the propyl chain to a pentyl chain. And so if you compare this compound to the parent compound, we actually, again, started to lose activity here, indicating that, while the propyl chain is really good for compound activity, if we go too far beyond that, we might start to lose some of the activity. In the synthesis of this BIN177 compound, we actually had an intermediate where we did not cyclize this last ring. And so we decided to make a derivative where we kind of kept this part uncyclized, so this region I'm highlighting in green. And we actually saw that this compound was slightly more potent than the parent. And so we decided to take this as kind of our core and expand upon this compound. And so again, we kind of went back to the same region on the left, those same two groups that I highlighted previously. And one of the first things that we did was remove that cyclopentyl group. And so again, we saw that we got a nice bump in compound activity, and this actually led us to one of our more potent analogs in this series, BIN178, which was now in the sub 10 nanomolar range. And as I mentioned, we had kind of been exploring what are the limitations if we go too far, extending longer than the propyl chain. We also made a derivative here where we shortened it to a methyl. And while this compound does, retain a lot of the activity, it's a little bit weaker and indicates that, that propyl chain is really in the sweet spot for size in that region of the molecule. And as I mentioned previously, we had done some experiments, some derivatization on this phenyl ring. And we had noticed that kind of substitution of 4 position with some of these larger nitrogen-containing heterocycles really seemed to kind of improve activity. And so we made a number of derivatives with some of the different scaffolds that we had with some of these changes, and saw we were getting compounds that were slightly better or comparable to this BIN168. And so we've been really driving down the potency for a lot of the compounds in this series. And so just to quickly kind of give a broad summary, our parallel HTS approach has really facilitated the rapid confirmation of hits as bona fide selective DUB inhibitors. These results provide insight into being able to accelerate the identification of some novel DUB inhibitors, and also potentially provide a roadmap for not just DUBs, but other enzyme families. Our medicinal chemistry efforts around the BIN10707 screening hit have identified some tractable SAR that really helps us think about the generation of best-in-class probes for elucidating USP28 function. And so we're continuing to work on derivatization of this series in the hopes of kind of further increasing potency, but also being able to fully understand what parts of the molecule are important for the observed compound activity. And obviously at this point, these compounds are targeting both 25 and 28. And so we're also starting to think about ways that we can engineer or kind of tune selectivity for each of the different DUBs through medicinal chemistry efforts. And so with our kind of best-in-class probe compounds at this point, we're really looking to start to validate a lot of the reported USP28 and potentially USP25 biology using our probes. We've started those processes with mixed success, but I think at this point, we're really starting to think about what are the contexts that we really should be using our compounds in. And those are all really important considerations for being able to kind of use these probe molecules to further elucidate the function of these DUBs. And so, as I mentioned, we're really trying now to study the different substrates, the function, as well as the therapeutic opportunities for both USP25 and USP28 using the compounds that we've developed from all of the work that we've done from the screen to the medicinal chemistry optimization. And so with that, I'll actually turn it back over to Sarah to give you a broad overview and summary.
Great. Well, thank you, Anthony. And I have just a couple slides here to, kind of wrap up some of the future areas that, we're excited about that has really been enabled by, Anthony's work. And, one of the real features, I think, of the screening and the follow-up that he's described that I just, wanted to note that I think is well aligned with the Target 2035 is really thinking about rigor, in these chemical probes and making sure that we really fully understand the potency and, importantly, the selectivity, of compounds that we're using to, study function. At the beginning, I introduced the idea of, we want to think about DUBs as a target class, much like people have done with kinases, make, use ATP mimetics, differentially functionalize to get libraries to identify inhibitors for many different kinases. And so, while in Anthony's talk, he focused on the follow-up around individual DUBs, more broadly, it's been very enabling when integrated with other inhibitors in the literature and some other screens that we've done to allow us to now design these DUB-focused libraries. And as I'm showing on this slide, these DUB-focused libraries now can span, 3 different pockets that have been previously described on DUBs. We're working on libraries for all of these. I'll just highlight, the first library that we really designed and profiled was targeting the catalytic site. These were covalent for the catalytic cysteine. And as we show on the next slide, even with this, first generation library, we've been, I think very productive in being able to identify hit compounds for a wider array of DUBs. So really by having, these focused libraries that we can screen broadly across, now it was 56 detected DUBs. These were endogenous DUBs. This was an screen done using activity-based protein profiling, which Anthony nicely introduced that technique. We have selective hit compounds for 23 DUBs. Although I acknowledge most of these are pretty, weak compounds. We're talking micromolar. We really need to do some subsequent medicinal chemistry optimization. But overall, I think this really bodes well for the opportunity to accelerate the pace of inhibitor development in the DUB space. And so just going on to the next slide, just to now summarize more broadly, today, I think we've shown that DUBs are an emergent drug class. There really is tremendous therapeutic potential across multiple indications. We've talked about inhibitors, but you can think about modulating these in many different ways. My lab has really invested heavily in establishing a platform to support DUB projects. Early on, this is really focused, on the chemistry and assay platforms. But, thinking about using these inhibitors for DUB function, we've succeeded in doing that for USP7, but we're also very invested in technologies so that we can understand function across, a wider array of DUBs. I think it's going to be really exciting to see, the future of this enzyme family. And really just to echo a really important point Ingrid made in the introduction, for these target classes where it's early days, the more we can work together, the more we can all benefit from accelerating progress. So, we are always happy to collaborate, whether that involves, sharing probes that are published from our lab or others, or, thinking about, a bigger collaboration focused around a particular DUB or a new technology. We'd be excited to, connect on any of those possibilities. And so with that, I think we are ready to open it up to questions, and please feel free to ask questions that are specific to some of the material in our presentation today, or about DUBs more broadly. And I can't believe I almost missed the acknowledgement slides. I was getting too far ahead of myself here. But Dana-Farber is really a fun place to do chemical biology research, to really be able to recruit very talented graduate students, postdocs, and work collaboratively with many of the groups, including some of which are listed here. And so with this, we really now will move on to the question. It looks like we have several that have already been typed in. Please continue adding them. And we also had a few that were submitted previously, so we will do our best to address those as well. All right, so maybe, oh, Go ahead, Ingrid.
No, I just wanted to thank you, Sarah and Anthony, for excellent presentation, very comprehensive, and then also, really focused on the details of, how important it is to do really careful optimization with developing deubiquinase inhibitors. So thank you for that. Yeah, so we can move on to the questions. Let's see. These are posted in the Q&A. I actually don't see any, so I'm not sure if anyone else is seeing any.
Yes, so I can see them here. So I can go ahead and start reading a couple of these. This first one is a specific question for Anthony. Did you test a molecule with piperazine ring and no cyclopentyl ring?
Yeah, so we do have some of those derivatives. I believe that one specifically we did make. And we did see kind of the expected SAR based on the SAR that I've shown you. But a lot of those compounds we're still kind of actively working on to synthesize and really fully characterize. But yes, we did make that compound, I believe.
Great. All right, here we have another one. Excellent talk. What has been your experience with using the ABPP with non-catalytic site inhibitors? Can you still see a decrease in probe binding?
Yeah, so that's an interesting question. I think at this point it's a little bit tough to deconvolute that because obviously, based on where the probe is binding, most of the time it's binding in the active site to that catalytic system. And so we're really just looking at active site inhibitors. I'm not aware of being able to, see probe labeling at non-catalytic cysteines. So I don't know if you have seen that previously.
Yeah, I was just gonna, chime in that when we say, non-catalytic site, we definitely will do the ABPP with ligands that they're not covalent for that catalytic cysteine, but they are still competitive with ubiquitin. And in that case, for some of those non-covalent inhibitors, our experience is that because you're competing a non-covalent ligand with a covalent probe, those compounds need to be fairly potent for us to really get nice results, in contrast to the covalent inhibitors where, even weak compounds, we can see a nice competition for that probe. We don't, I think, have specific experience with compounds that would be binding non-competitive with ubiquitin, but have inhibitory activity. I would think ABPP should still work, but I can't cite a specific example. All right. Okay, Anthony, interesting that USP17 had so many hits. Any thoughts on why this is the case?
Yeah, so part of that is a little artificial in that the primary screen we screened at 50 micromolar instead of 25 for the other. So most of those hits, I'm guessing, are things that are maybe just aggregating or somehow other affecting the assay when we're testing at that high of concentration. But for USP17, we saw that most of those compounds were actually kind of ruled out when we went through our dose response. And so my guess is that's most likely where we're seeing kind of the large number of hits for USP17 there, but not really much when you do the dose response follow-up.
And I apologize, I was kind of reading the question, so I might have missed it, but we also saw that a large number of those hits for some reason were natural products. That library had a small couple thousand compounds that were natural products, and they weren't really tractable for us, for follow-up. So that was another. Okay, so another question here, you showed that the HTS was done in cells. I was wondering what type of cells, because this can affect the results. Was the cell type connected to diseases mentioned in the second part of the talk? So maybe I'll answer this one. So for the high throughput screen that Anthony was talking about, that was done with purified doves. And then for some of the target engagement work, that was done either in HEC293 cells or some cell lines where, there was reported disease relevance. So exactly as you show, we wanted to make sure that when we were looking kind of downstream at function, that we'd shown target engagement in the relevant system. I highlighted just, one of the last slides that we have, been trying to move with our focus libraries towards screening, at least in cell lysate. So, a bit more physiologically relevant, using the ABPP. In that experiment, we're starting with HEC cells, but, then always doing follow-up studies in, a cell line of interest that is relevant. So hopefully, that kind of addresses, the question. Certainly, maybe an extension of this kind of line of thought would be phenotypic screens, where we now can take these inhibitors and, profile for a phenotype of interest in a disease relevant setting, cell line, organoid, whatever the case may be. And then think about, going in and looking at the selectivity data that exists for, any hits to, kind of build projects from that angle. Let's see. Let's see, thank you. Oh, here's one we can add. Is there any chance of making the raw data for the original high throughput screen publicly available for others to mine and ID selective subtype inhibitors? Anthony?
Yeah, absolutely. So as part of our kind of collaboration with Novartis, all of our primary screen and dose response data was uploaded to PubChem. So it is all publicly accessible in PubChem. You can get the primary screen data as well as the dose response data for all the compounds that were tested as part of this collaboration.
Let's see. How much computational chemistry helped your DUB screening? And so, I will, I guess, let's see. So that probably, we can answer maybe in the context of some of our library work that's been a follow-on from the initial, high throughput screening. The truth is at this point in time, a lot of our computational work is still focused, say, on a single DUB. We know we have an example for a particular scaffold where it hits, 28, a couple other DUBs, and we've, been able to use computational approaches to, generate models of those compounds bound to that DUB. But I think there's still a lot of potential for being able to extrapolate that data to start to make predictions about other DUBs that might be targetable, with that scaffold, and to, introduce more rational design. So the truth is we're starting to implement it. We're finding that it's definitely beneficial, but we also, truth be told, recognize that there's really, I think, a lot more that we could do in terms of computational studies, as well as, say, machine learning, with some of our, existing data sets. And I think we have a large enough body of data now that it's a good time to really start expanding our expertise and collaborating, in that direction. Let's see. Do you have any compounds with USP25 activity, but no USP28?
I wish. At this point, we haven't seen that. We have been testing our compounds against 25 and 28. We haven't seen any compounds with selectivity for one over the other at this point, but that's still something that we're following up on to try and see what we can find.
Let's see. Okay, let's see. Your ABPP target engagement assay was performed in cell lysates. Do you know how cell permeable your inhibitors are?
Yeah, so at this point, we haven't really done too much to look at that. One thing that we could do is, live cell target engagement. So incubating the compounds in actual live cells and then doing the same thing to see, if they actually get in. And so we don't really have any evidence yet to kind of point to the fact that they do or don't get in, but that's something that is kind of coming in the pipeline for a lot of these compounds, especially our really, really potent.
Let's see. So then there is, a question about, biophysical assays with the USP28 to as well as some other kind of mechanism type studies to establish whether these are, competitive with ubiquitin, binding another pocket. Anthony?
Yeah, so we've done a little bit of kinetics for some of our kind of favorite compounds. They do appear to be competitive with ubiquitin based on those results, but we are still kind of thinking about additional validation experiments and additional orthogonal experiments to be able to really fully characterize those types of interactions.
Let's see. So we also have, a few questions here that were submitted beforehand. One is, a specific question, but maybe I'll generalize it a little bit. The question is, are there any deubiquitinases that could be considered as drug targets for substance abuse disorders? DUBs have been implicated broadly in many different therapeutic areas. The two DUB inhibitors that are entering the clinic, one is it's in cancer for BRCA deficient tumors. The second is for chronic kidney disease. There definitely is a fair bit of literature linking DUBs, to neurological disorders as well as infection. The viral DUB PL-Pro is certainly getting a lot of attention these days because it's encoded by coronaviruses. So to answer the question specifically, I'm not familiar with any DUBs for which there is a strong link, to substance abuse disorders, but there's still so much to learn, about their function. And I do believe they're going to be, broadly involved in different, therapeutic areas. We also have another kind of question here that I'll maybe modify just a little bit. Are there available computational modeling methods, thinking about bifunctionals in this ubiquitin space? And so this is relevant, of course, to, the PROTAC, the compounds that are going to be recruiting E3 ligases to degrade a protein of interest, but now also for DUBTACs, which were recently, published by Dan Nomura, where you can recruit a DUB using a bifunctional compound to achieve targeted protein stabilization. I will admit this is not fully my area of expertise, but we've certainly enjoyed, collaborations with, Eric Fisher here, for example, on being able to model ternary complex formation when we know the binding mode of ligands to both of the proteins of interest, to get an idea of a linker length that might be optimal, what types of productive interactions there might be between, the two protein binding partners. So I think that there's a lot of opportunity, in that space, really to be able to design your initial bifunctional compounds, but then, also to refine as you move forward. And it looks like we are coming up kind of at the end of time here. So I think perhaps we should go ahead and wrap up.
Yeah, that sounds great. So thanks everyone for attending. Thanks for all the great questions. And most of all, thanks to our speakers, Sarah and Anthony. Really spectacular overview, and then also deep dive into the importance of really careful characterization of developing deubiquitinase inhibitors.